Hello everyone. My name is Madhvi Singh, Assistant Professor from IIMT College of Pharmacy. Today we are discussed about gene therapy. So there is the points we are covered in this lecture is genome, genomics, components of the human genome, gene therapy, history, types of gene therapy, vectors involved in gene therapy, advantages and disadvantages of gene therapy. So first is what is genome? The genome is all the DNA in a cell. All the DNA on all the chromosomes includes gene, intergenic sequences, and repeats. Specifically, it is all the DNA in a organelle. Eukaryotics can have 2 to 3 genomes. First is nuclear genome, second is mitochondrial genome, and third is plastid genome. If not specified, genome usually refers to the nuclear genome. Next is genomics. Genomics is the study of genomes, includes large chromosomal segments containing many genes. The initial phase of genomics aims to map and sequence an initial set of entire genomes. Functional genomics aims to deduce information about the function of DNA sequences. Should continue long after the initial genome sequences have been completed. Next, what is human genome? Human genome consists 22 autosome pairs plus 2 sex chromosomes, 3 billion base pairs in the haploid genome, where and what are the 30 to 40,000 genes, and is there anything else interesting and important about human genome? So what are the components of the human genome? Human genome has 3.2 billion base pairs of DNA. About 3% codes for proteins. About 40 to 50% is repetitive, made by retrotransposition. So what is the function of the remaining 50% genome? What is gene therapy? An approach of treating diseases by either modifying the expression of an individual's gene or correction of abnormal genes. This is accomplished by 3 steps: replacing, inactivating or knocking out, and introducing a new gene. First is replacing. Replacing a mutated gene that causes disease with a healthy copy of the gene. Next is inactivating or knocking out a mutated gene that is functioning improperly. Introducing a gene into the body to help fight a disease. Next is history of gene therapy. In 1960, the concept of gene therapy was introduced. After that, in 1984, a retrovirus vector system was designed that could efficiently insert foreign genes into mammalian chromosomes. Then, in 1990, the first approved gene therapy in the US took place on 14 September 1990 at the National Institute of Health under the direction of William French Anderson. After that, 4-year-old Ashanti De Silva received treatment for a genetic defect that left her with ADA-SCID, a severe immune deficiency system. In 1999, that of Gelsinger in gene therapy experiment. Okay, then is types of gene therapy. First is somatic cell gene therapy and second is germline gene therapy. So let's here discuss about somatic cell gene therapy. This gene therapy includes therapeutic genes transferred into the somatic cells. Examples: introduction of genes into bone marrow cells, blood cells, skin cells, etc. It will not be inherited lateral generation. This gene therapy means somatic cell gene therapy, not be inherited lateral generation. Next is germline gene therapy. In this therapy, therapeutic genes transferred into the germ cells. Example: genes introduced into eggs and sperms. This germline gene therapy is heritable and passed on to the lateral generation. Next is types of somatic cell gene therapy. Two types of somatic cell gene therapy. First is ex vivo gene therapy and second is in vivo gene therapy. So what is ex vivo somatic cell gene therapy? Cells are modified outside the body and then transplanted back in again. It is called ex vivo because the cells are treated outside the body. Next is in vivo gene therapy. In this, genes are changed in cells when the cells are still in the body. Called in vivo because the genes is transferred to cells. Next is vectors in gene therapy. To transfer the desired gene into a target cell, a carrier is required. So these carriers, such vehicles of gene delivery are known as vectors. Vectors has two main classes, viral vectors, non-viral vectors. Viral vectors. Viral vectors, in this, viruses introduce their genetic material into the host cell as part of their replication cycle and then remove the viral DNA and using the virus as a vehicle to deliver the therapeutic DNA. The viruses used are altered to make them safe, although some risk still exists with gene therapy. So here is the diagram in which cells with original genes, after the exposure, viral vector carrying new gene, then vector insert new gene into the cell. After that, the new gene in the cell along with original genes. You can see there. Next is types of viral vectors. First is retrovirus vector system. In this system, the recombinant retroviruses have the ability to integrate into the host genome. It can carry a DNA of size less than 3.4 kb. Target cell dividing. Next is adenovirus vector system. Adenoviral DNA does not integrate into the genome and it is not replicated during cell division. In humans commonly come in contact with adenovirus, majority of patients have already developed neutralizing antibodies which can inactivate the virus. This is the diagram showing the gene therapy using an adenovirus vector. So vector bind to cell membrane, then modified DNA injected into the vector. After that, this process vector is packaged in vesicles. Vesicles break down, releasing vector. Then cell makes protein using new gene. Vectors injected new gene into nucleus. Then this is the new gene and vector adenovirus. Next is non-viral vector system. Pure DNA construct. In this, direct introduction of pure DNA construct into target tissue, efficiency of DNA uptake by cells and expression rather low. Consequently, the large quantities of DNA have to be injected periodically. Human artificial chromosomes. It can carry a large DNA with one or more therapeutic genes. In the essence, with one or more therapeutic genes. Next is the advantages of the gene therapy. Gene therapy has the potential to eliminate and prevent hereditary diseases such as cystic fibrosis, ADA, SCID, etc. It is a possible cure for heart diseases, AIDS, and cancer. It gives someone born with a genetic disease a chance to live life. It can be used to eradicate diseases from the future generation. So, apart from this, there are various ongoing researches, further researches which are ongoing on gene therapy. Next is disadvantages of gene therapy. Long-lasting therapy is not achieved by gene therapy. So this is the disadvantage. Due to rapid dividing of cells, benefit of gene therapy is short-lived. And next is immune responses. Immune responses is also a disadvantage in gene therapy procedures. In this, immune response to the transferred genes stimulate a potential risk to gene therapy. Disorders caused by defects in multiple genes, diseases and disorders and any kind of illnesses जो caused हो रही है by defects होने से in multiple genes cannot be treated effectively using gene therapy. Viruses which are used as a vectors for gene transfer may cause toxicity, like examples हमारे adenovirus, retrovirus, immune responses and inflammatory reactions in the host. These are the disadvantages. So this is all about the gene therapy. Thank you so much.